All right, good afternoon. I want to finish what we started and finish off chapter 11.3, just the parts that have to do with transport that relates exactly to enzyme kinetics. So let's go ahead and start, and I want to review where we started with the dancing squid dish, where you add soy sauce to a squid and the salt makes the nerves twitch, makes the squid dance on your plate. So we talked about how there are transporters that take the salt into the cell, and that is somehow part of the nerve cell. It goes through a protein like this. We have a protein with a specific active site. We have a starting concentration and ending concentration. It's just we're talking about two places rather than two configurations of bonds. But that works with the equations. And so I want to say that we have these voltage-sensitive sodium channels that get the salt into the cell, and it's a fundamental part of how nerves fire. Now, the one thing that we have to talk about: how do you talk about a free energy moving between two spaces? So you have two spaces separated by some sort of barrier that the things can move across however slowly or however quickly. If you have that kind of barrier in place, then you can measure the rate of diffusion across of it, because if you have a case like you have a disequilibrium across those, where you have more on the left in C1 than on the right in C2, then you will start off with a situation that's different than what you'll end up with. That situation will move, is far from equilibrium. It will move to be at equilibrium, where literally both of the concentrations are equal. This is true for uncharged solutes, where the only thing that you have to equalize is the concentration. The concentration will have a force that pushes it toward equalization. Technically, it's kind of weird for me to even talk about it as a force, but it's effective. It's an effective heuristic if you want to talk about philosophy and things like that. In fact, what this is very effective for is we can talk about there's more of a force if there's more of a concentration difference on each side, because there's farther for it to go, and it will move faster, at least at first, toward the equilibrium situation. At equilibrium, delta G equals 0. Also, delta S between the two sides equals 0, because the solute is spread out evenly on both sides. Before equilibrium, delta G, for moving in the direction that it wants to move, is going to be negative, because that's what happens with spontaneous reactions. And the delta S of moving from C1 to C2 is going to be positive, because you're going to have the solute spreading out across the membrane. So you're probably familiar, if you've encountered this concept before, as the force of osmosis, osmotic pressure, which will cause cells to lyse if you put them in water, because you have the salt causing huge concentration gradients across the membrane that disrupts the membrane. I want to say that you might have been taught some aspects of osmosis wrong. And if you're interested, it's not just a simple entropy effect like this, although we can start and we can describe our enzymes using this. If you look into it in detail, you actually see some problems with this explanation that don't show up in this class. And I just have to mention this because I think this is fascinating when we find something that is not quite the way it's been taught. Five popular misconceptions about osmosis is a paper that discusses that. You can look that up from the bottom if you're interested in this. And sometimes if students are really interested, we talk about it in Biochem 3, 4363 in the spring quarter. If you're one of those students, let me know, because I think it's an interesting problem. But definitely, we're not talking about mechanism. We're not talking about how. We're just talking about what. And what happens is that concentrations have a force to equalize through a membrane. And so even if you have equal concentrations across a membrane, you know, the force to equalize is the delta G of reaction. So it has to do with the reaction between the left line and the right line. Here's a case where you even have two equal solutes across the membrane, two equal concentrations, where you have the left line and the right line being exactly equal. Even in that case, though, there's a barrier to get through if a solute is going to move from one side to the other. And there's two parts of that barrier that's shown on this. If you have, depending on the nature of the solute, it's going to have a tough time. If it's easy to dissolve in water, it's hard for it to dissolve in oil. And the membrane is oil, so that means that its free energy when it is in the membrane is going to be very high. Not infinite, but very high. And so it's going to be slow for it to move across the membrane in any way. There's also even a little extra barrier to it. If you look at this on the red line on either side, the red line shows that there is an extra barrier that has to happen, because to be in the middle of the membrane, you have to strip off all those waters. You have to desolvate the ion, which means you have to peel all these polar charges away from the hydrated solute, however they're dissolving and interacting with it. And that's another energy barrier. That's the little energy barrier on the top that you see on either side of the membrane here. Either way, this is a barrier that slows down the process of the solute moving across the membrane. However, if we know if there's a high energetic barrier, if we just bind the transition state, in this case bind the solute in the middle of the membrane, we can bring down the energy of that. And so it doesn't even have to be brought down all the way. If we just bring it down significantly, then it's easier and faster for the solute to transport across the membrane. And that's what it's showing right here. This looks very similar to the diagrams that we showed of enzymes binding the transition state. Transporters also bind the transition state, and they do a conversion in space from substrate inside to substrate outside, or vice versa. So this is true, and this is true even if delta G is 0. Even if you're at equilibrium, technically the barrier is still there. Even more so if you have a difference between the delta G of the beginning and the ending, then you'll be able to reach that equilibrium or be able to drive across it more quickly. So the solvation that is shown here is the resolvation is another barrier that is equal and opposite to the desolvation on the left side. So we're going to start with a solute that is uncharged. The only thing we have to think about is its concentration. And there's a transporter for glucose. Glucose is hugely important, and it flows through the blood, and then it gets transported into cells. So this is a hugely important process. There's a transporter for it called GLUT1, and it's pretty simple. It's actually just 4 alpha helices embedded in the membrane. And what they are, if you look at the helix wheel, here's again a concept from earlier in the class. If you look down the helix wheel, you have one side of the helix that is polar, and all the other sides are nonpolar. So they will interact well with the membrane, but you will have this sort of, if you're looking down on the membrane from this perspective, they just have this polar hole in the middle of the membrane. And so they bind the glucose halfway through the membrane, bring down its energy, bring down the barrier to the glucose crossing the membrane. And you have a glucose transporter. You have that helix wheel 4 times around to make a little pore in the middle. So here's an example. Here's a bacterial ortholog of glucose transporter, which is it actually transports a different sugar, but it's the same idea. You have, in this case, you have a number of different helices embedded in the membrane, and you can see that these helices actually sort of are tilted in so that the hole in the middle of the membrane is closed, unlike the sodium channel that we showed before. But these can also move, and so they can actually tilt in and out so that that little V shape, that black V shape on that side, can actually open up to allow the glucose in. There's a picture of the glucose stuck in the middle, sort of exactly at the transition state. And in fact, there's another thing that has to happen for this enzyme to catalyze the reaction. The enzyme itself has to undergo a conformational change. This is one of the ways that you can talk about the way that glucose can do that. And so the glucose binds while it's closed on the inside, and then it closes on the outside and opens on the inside. Step 2 is 3. It releases the glucose. That's like product release. And then it returns to the original configuration. we can talk about the energy of all this if we just are looking at the concentration difference between the inside and the outside. That's the delta G of reaction, and then the enzyme will change the delta G to the transition state. And so we can talk about this situation with 4 arrows. And these look exactly like the four states, I should say, with at this point 6 arrows. But we can do everything that we did with Michaelis-Menten kinetics. Instead of having substrate to product, we have substrate out to product in. Instead of having enzyme, we have transporter, so we call it T, but it works exactly the same for all the equations. And instead of Km, we just call it Kt because it's a K for transport. And all the math is exactly the same. So the good news is the math that you are working on learning is applicable to multiple situations. And in fact, if we can do all that, then we can get rid of the same arrows, we can rearrange, we can write the same equations. And so the same equation gives us the same graph. So we have a Kt at half Vmax. We have a Vmax, that name doesn't change. And we have, instead of S, just plain S, in this case we have S out, which is we're adding the glucose on the outside of cells, so that's our substrate, and our product is how much glucose gets in the cells. And so we can even do a straight line, a Lineweaver-Burke plot, and we can even do inhibition. Everything is the same. We're just changing S to S out. And we can also do the likewise if we're looking at transport from inside to out, then we change it to S in. All the labels are transformable. So there's Kt. And so instead of measuring -1 over Km with the x-axis that we talked about before, now we're doing -1 over Kt. The only extra equation you really need is not really a graph, it's more of a calculation. Because we can have a delta G, the delta G for a reaction is products versus reactants. So the delta G for a transport reaction is products versus reactants by the same thing. And the only thing about that is that you actually can use the math to relate the delta G directly to the concentrations by using this equation right here. Delta G of transport equals RT natural log of C2, which is the product concentration, over C1, which is the reactant concentration. Products over reactants, just like always. So actually what you have inside the natural log parentheses is kind of like an equilibrium constant. In fact, it is an equilibrium constant for the transport reaction. So for example, if you see, let's say that if C2, if you're trying to move a molecule from a place where it is 100 times more dilute to a place where it is 100 times more concentrated, then that's going to be hard. The delta G for that is going to be positive. The natural log for that ratio is going to be positive, natural log of 100. And then the delta G of transport is going to be 5.7 kilojoules per mole. I believe that's at room temperature. But just notice that you need the temperature to really do the calculation for this. But you can get a relatively large positive thing from moving something into the cell. For example, for glucose, usually the concentration inside the cell is 100 times the concentration outside. So you need some help to do that. And so you can do calculations, and we have calculations like that in the homework. The cool thing is that because the enzymes bind the transition state, they can do it slightly differently. They can do conformational changes slightly differently, and that will come out in having different Kt values and different Vmax values. So you can measure the different Kt values, and it turns out that there's lots of different tissues that have different glucose transporters with different Kt values. So you have these, and you look at these, look up and down and see where the lowest Kt values are the tissues that need the most glucose. They will transport more glucose in at the same concentrations, all other things being equal. And if you look up and down, there's a large diversity in these things, but you can see that there's a brain, like GLUT3 is a brain transporter that has a lower one than the ubiquitous GLUT1. you can think about why these transporters need to be more efficient and they need to bind their solute better. So you can compare the ubiquitous one, which is about a 3 millimolar kT, and to the brain one, that will be a 1.4 millimolar kT. Okay, so that's what you can do, and if you need to move it in against a concentration gradient, then you need to have some kind of negative delta G. You need to have some burning ATP or some other reaction you can couple to it that will have a negative delta G. So, what if the solute is charged, however? Because if it's charged, we have to think about something else. The charge across the membrane also affects where the equilibrium is. So if you have just all the same concentrations, but if the charges are different and you have multiple pluses on one side of the membrane and multiple minuses on the other side, you have multiple minuses on the left here and multiple pluses on the right, well then, to reach equilibrium, there will actually be a driving force that will cause equilibrium to happen when you don't have a charge difference anymore across the membrane, when you have a perfectly mixed amount of pluses and minuses, as well as a perfectly mixed amount of the solute itself. This is expressed as a voltage, which just depends on the charge imbalance. So now we're talking about a separate component to the energy that only kicks in if you have something like salt. That is a charged solute. It does not appear for glucose, which is not a charged residue. But you express that as a voltage, and you express it in millivolts, and at equilibrium, the voltage across the membrane is 0. Before equilibrium, the voltage is positive, and that means there is a driving force to move toward 0. So this means that we have another component in the energy to think about if we're going to understand what's going on with the driving forces that are bringing salt into the cell. If the solute is charged, you have to account for the charges as well. That means that we have the same equation we had before, RT natural log C over C, plus another term that accounts for the delta G that is due only to the charges, and this depends on voltage. So this sounds really complicated, and it does complicate things a little bit. But it's not that bad because it's really just 3 things to multiply, and then you add it in and you see what the delta G overall is. The Z is just the charge on the ion. For sodium, it's +1, so it's going to be a Z of 1. The fancy F is Faraday constant, which is just a constant relating joules to voltage. And so it makes everything cancel out really nicely. Just make sure your units are canceling out. Really, the only thing you have to plug in is you have to plug in the transmembrane electrical potential. This can literally be measured by having electrodes inside and outside the cell, or there are other ways to get at it as well. But it is literally the voltage across the membrane, which will affect how positive and negatively charged things are pulled or pushed across the membrane. Normally, the delta phi, which is the transmembrane potential, is 50 to 200 millivolts. See how I have it in slightly different units on the slide. And usually the charge is more negative inside the cell. The reason why the charge is more negative inside the cell is because cells are alive, they live at disequilibrium, and they work a lot, they burn a lot of ATP to be at disequilibrium. One of the most important disequilibriums in a cell is that a live cell will have more negative charge on the inside than is outside the cell. The reason is there's this protein, the sodium-potassium ATPase, which is an active transporter that is an antiporter for sodium and potassium. And so it burns ATP to move away from equilibrium. So it's pumping you away from equilibrium, and it ends up constantly burning ATP to bring 2 potassiums in and 3 sodiums out. If you look at that, that means you set up a disequilibrium where you have more potassium inside a live cell than outside, and less sodium inside the live cell than outside. So that creates this voltage that is across the cell. You measure the voltage, you plug it into your delta G equation to see what your delta G is from moving your solute into a live cell. So neurons really depend on this. I believe all cells have it, But neurons really depend on this because neurons are constantly moving salt in and out of the cell as they fire. And this pump then has to work to sort of reach the disequilibrium that they do. If this pump is working, that means that the firing of the neuron is actually passive, because you're just opening up sodium channels and letting the ions flow down their gradient. This is why the dead squid still has this voltage across its cells, and the dead squid neurons will still fire in response to salt, because they are still at disequilibrium. They've kept this imbalance in place, even though they aren't actively pumping anymore. And this is really important because 25% of all resting ATP consumption is used for one protein. Here's a quote from Bill Bryson. At any moment, typical cell will have 1 billion ATPs in it. In 2 minutes, every one of them will have been drained dry and another billion will have taken their place. So every day, if you calculate this out by the number of cells, you produce and use up a volume of ATP equivalent to about half your body weight. I could quibble with this a little bit because you're really just forming and unforming bonds, but go with him to just say how much you do this. If you feel the warmth of your skin, that is ATP at work. Generally, that is actually transmembrane potentials, and you're burning energy to stay warm. You're literally like having little fires going on that are burning ATP. So the ATPase demo with a little help. Here's a little help from Drake. He had a video where he did this movement, and he had 2 movements to the right, 3 movements to the left, and then he kicks. And that's actually what the sodium-potassium ATPase does. It just does it all at once. We're not talking about mechanism, but it moves 2 potassiums into the cytosol. It moves 3 sodiums extracellularly, and to pay for this, because this takes energy, you're moving it against the concentration gradients, you use ATP. The negative delta G from ATP pays for the positive delta G of doing this process. So, in terms of overall ATP and energy use, we're going to have the sodium-potassium ATPase is shown in gray, and so you use a lot of it, and also the corals use a lot of it as well. And so you have CO2 is changing. When you change CO2, you change the way the corals are using their ATPase because you're stressing the cells, and it shows up in this exact pump activity. The gray value is shown for these sea corals. I don't even have the species right, but what matters is it's a totally different animal. When they're under stress from having CO2 high levels of that, ocean acidification from global warming, the gray values go up. Look particularly at part B, and look how the larvae and the control have, they're using 20 to 30% of their ATP for this one protein. That goes up when there's a lot of CO2 to be 45%. That's doubling from having the more stress from the CO2 because they're having to fight more to keep their homeostasis in this different environment from what they evolved in. So let's just say if you've heard that ocean acidification is bad, this is one of the reasons, because they have to fight harder to keep alive, to keep their cells firing, and especially neurons. This is what it looks like. We can't get into the structure really, but these are really amazing structures. How does it pump 3 sodiums out and 2 potassiums in? Where does the ATP bind? That can all be other discussions for another day. Technically, you can get into that. But the main thing I want to talk about is that we can have drugs that are inhibitors of this. And so here's this wabane, which is this molecule that it looks a little bit like sugar. It's kind of weird, but however it does it, this binds and inhibits sodium-potassium transport. And it's from foxglove, which is why foxglove is actually poisonous. What's really weird is that this drug that blocks the sodium-potassium ATPase is actually, it will kill you if you take it under the wrong circumstances, but if you give it under the right circumstances, it will actually kickstart your heart. The reason is, it's a little biologically complex, but if you block sodium-potassium transport, there's a backup system that is a sodium-calcium antiporter that will actually get calcium into the cell. And so you'll be stressing the cells, but they'll get calcium into the cell, and that kickstarts heart muscle contraction. So I don't know how they found this out in the first place. It's a pretty amazing mechanism, but this is a sodium-potassium inhibitor that will kill you under certain situations, but it will save your life if you're having a heart attack. And that's what it looks like. There's one other thing to talk about. It's called ionophores, and this is another kind of drug. This is not binding into as a competitive inhibitor, but this is offering another kind of transport to get things across the membrane. So for example, Valinomycin is an antibiotic that binds potassium ions and is able to transport them across the cell, and that will kill bacteria, because the bacteria are supposed to be keeping all their potassium inside the cell. Well, if this basically opens up a hole in the membrane, not literally, but practically speaking, it opens up a hole, it basically allows the potassium to move across the membrane, because it's got all this hydrophobic on the outside, and it's got this little potassium-shaped hole on the inside. So if you add this to the bacteria, it will move them across the membrane, and they won't have enough potassium on the inside, they won't have the transmembrane voltage, and the rest of their transporters won't work. It's good news that this doesn't work on our membrane. Our membranes are sturdier against it somehow, but it definitely does a good job against bacteria. So here's an example of another small molecule that actually literally forms a channel in the cell. In the normal case, you have a proton channel shown in purple, and then you have a potassium channel shown in green. And so the potassium channel lets the green potassium across. The proton channel is letting protons out, and normally you have a balance against that in a normally growing cell. If they mutate in yeast, they mutate the potassium channel, then you have the purple still pumping out, but you have an imbalance because you don't have the potassium going in. And then you have an imbalance where you have more positives on the outside than you should, because the outside pumping is working, but the inside pumping is broken. In this system, if they add Amphotericin B, it will actually form a channel. That little molecule will form a channel in the membrane that will let the potassium back in and serve the function of the potassium transporter and let the cells continue to grow. So how does the Amphotericin B work? It actually makes a big ring, and it makes a hole in the membrane that is specific for potassium and allows the potassium to go through. This is how easy it is to make a transporter, and in this case, the potassium moving down its transportation gradient is actually something that the cell wants to happen. It's passive transport, and this will actually allow the cells in that place to grow. It will replace their broken potassium transporter. So anyways, those are the stories about the transporters. I want you to work through the homework, realize that there are many different cases. All 6 of the cases that we can talk about are shown on this slide. But all of these cases can be talked about as enzyme reactions. And that's what I've got to say about Chapter 11. So now work through your homework problems involving inhibition or transport. Hopefully you're watching this over the weekend, and I will also give you an assignment over the weekend to get started on Chapter 7. But this is it for Chapter 6 and for Chapter 11 for now. We'll get back to Chapter 11 in the next test unit. For now, just work on the homework and let me know how it's going. We'll review it on the test day. All right? Have a good weekend, everyone.